Howdy, this is Yu-Sheng. Today I'm going to present you a paper, End-to-End Differentiable Learning of Protein Structure. This paper is from Cell System 2019. Let's talk about the main topic. The main topic of this paper is to use a novel method for predicting protein folding. This is a sequence to structure task. This task is to determine the spatial arrangement of atoms within a protein molecule. The significance of this task is that it is important for understanding protein functions and thus can help drug discovery and design. However, existing prediction methods are human engineered, and they have other issues which I will describe in detail later. This proposed method introduces an end-to-end differentiable deep learning model for predicting protein structure from sequence. And it claims that it utilizes geometric units that can optimize global geometry without violating local covalent chemistry. What is thrilling is that this model achieves state-of-the-art accuracy in predicting novel folds and pretty close to the state-of-the-art for known folds. Before this method is proposed, let's see about computational methods for protein structure and prediction. So these the methods basically fall into two main categories. One is explicit sequence to structure mapping. This involves physics-based simulations and fragment assembly methods, and relying on experiment structures as templates. As we can see, since they are using physics-based simulations, they are easily restricted by computational cost to small proteins. Another category is co-evolutionary methods. They identify co-evolving residues within protein families to derive contact maps, aiding in structure prediction. However, this method is unable to predict structures for which no sequence homologs exist. In other words, the novel protein design may not be suitable for this kind of methods. So the proposed recurrent geometric networks, aka RGNs, represent a promising approach in this domain. It is able to predict the novel protein design and not too restricted to computational cost. Let's see how this approach is designed. This is the overview of the model. As we can see, this is the input of the model, which is a protein sequence. And the overall output is the predicted structure of predicting structure fold for this protein. In this box is the model itself. We can see that it consists of two kinds of units. One is computational units, and another one is geometry units. Computational units is essentially a bidirectional LSTM, which can integrate additional information on every single unit, or say amino acid or residuals. Since this is a bidirectional LSTM, it can integrate neighboring amino acid information to a specific amino acid. And this computational unit will output torsional angles, which is a predicted torsional angles, to the geometry unit for further computation. The geometry unit is taking each component, or let's say each residual, and additionally adding more and more units onto the backbone, onto the partial backbone. So as the partial backbone grows, it eventually will give the predicted structure as the output. In this box, we can see the predicted structure and the experimental structure. Want to compare it together and calculate a loss for the deep learning model, for the backpropagation of deep learning model to learn, to update its weights. We're using DRMSD as our loss function. We'll get into the detail later. Back to the detail of the computational unit. As I mentioned, it is basically a many-to-many bidirectional LSTM with 3 output angles. Here, we look into a special component here in the computational unit. As I mentioned, this computational unit is going to output 3 output torsional angles. And this torsional angles is decided by torsional alphabet, which are some learned torsional angles, some combination of torsional angles, some learned. And we put it into a circular projection, and we use a weighted average to calculate the output torsional angles. What is the weighted average? How is the weighted average decided? By the LSTM. LSTM will learn what the weight will be. Back to the geometry unit. So what geometry units essentially do is that it takes an existing input, existing atom as an input, do some bond rotation, bond extension, some torsional rotation. Basically, it is trying to adjust the coordinates of the atoms. Back to the loss. The loss function DRMSD, in a higher level overview, we can view it as the distance, the so-called distance between the predicted structure and the experimental structure. Experimental structure is basically just the ground truth. So how is it calculated? First, we calculated pairwise distance within a structure. And then we have the experimental, the distance, the pairwise distance of the experimental structure and the pairwise distance for the predicted structure. And since it's a pairwise, we're using a specific element and calculate that for that specific element, the distance between the experiment and the predicted. So we have a matrix that is D. So the element of D, of capital D, is this DJK. And since it's a matrix, how do we represent the whole so-called distance between predicted structure and experimental structure? We take the L2 norm of this matrix. However, since the structure can be different length, so we take L times L minus 1 as a normalized term. Let's get into the results, experimental results. So for the experiment, we have to assess the model. So how do they do? They follow the critical assessment of protein structure prediction, CSP. This is a kind of procedure if they evaluate a model. What kind of procedure? It assesses models in a blinded fashion by testing picture using sequences of soul structures that have not been publicly released. So basically, it's like a testing in machine learning term. The evaluation matrix, one is the loss function we were using, DRMSD. Another one is TM score. This is because DRMSD assesses predictions at all length scales, and it penalizes large global deviations in proportion to their distance, resulting in very high errors for far apart regions. That's why the authors of the paper introduced TM score into play for that. And also, DRMSD is invariant to reflections. So the model, when using DRMSD as loss function, the model might have to utilize some correction, because it might generate reflected reflected 3D structures which are not reasonable. This is TM score. I'm not going to the detail of the TM score. We have to know what we have to know is that TM scores range from 0 to 1. The larger, the better. A standard is that if the TM score is less than 0.17, it is basically telling you that two protein folds, they are randomly unrelated. If the TM score is larger than 0.5, it generally corresponds to the same protein fold. It's basically saying that the two protein folds are really alike. Also, it is not invariant to reflections. The data set is recreated by CSP condition by assembling the protein net data set. So it is not directly using CSP, but they are using protein net dataset in a CST fashion. Also, it utilized multiple CSP datasets for that, and it adopted CSP division of test structures into two kinds. One is free modeling, one is template-based modeling. And what free modeling or FM, it is about predicting novel folds, and the other one is about known folds. Predicting novel folds with known homologs using protein data bank dataset. So we can say that the experiment is using two datasets. One is protein net dataset. Another one is protein data bank dataset within using a CSP dataset format or fashion. This is the experiment results using DRMSE as the metric. So RGN is we are comparing our RGN with the 5 candidates. What are those 5 candidates? There are servers using evolution methods. So as we can see, for novel folds, RGN outperforms all of them. But in null folds, it still falls behind the best server for CSP. However, we should note that the difference between the result of RGN and the best co-evolution method based servers are less than within 1 to 2 or, which is not quite unacceptable. For experiments using TM score, we can see that the TM score of RGN is not very good. It is in the 3rd and 4th, between the 3rd and 4th rank for novel folds. However, in known folds, and in known folds, it's also not performing very good. However, we have to note that the TM score of the known folds is really close to 0.5. As I mentioned, 0.5 is generally correspond to the same protein fold. So this is actually not so bad. To conclude, RGN showcases promising advancements in protein structure prediction, and it outperforms existing methods in predicting novel folds, and are close to state-of-the-art, which are co-evolutionary servers in known folds. Yeah, as we know, it's evaluating using DRMSE. And the best part is that it does not have to rely on co-evolutionary data or structure templates, demonstrating its robustness and potential. Future directions may involve, incorporating additional information such as structural templates or co-evolutionary data. My personal critique is that this method could integrate some physics-informed mechanism in the LSTM-like model. I think it may help further restricting the output result and make them more realistic, which might help solving the reflection problem, as I mentioned before. And another thing is that even though they claim to avoid violating local covalent chemistry using torsional alphabet, I personally not convinced since this is just choosing from a list of correct states, but it does not really ensure the physics rule in general. And this is end of my presentation. Thank you so much.